I'm going to share my screen. All right, hopefully you can see my screen. What we're doing today is Lab 5's streaking for isolation and culture media. In the lab, you would actually be streaking out plates, but you can't come and do that, and so we'll show you little videos of it, and I think there's a virtual lab for this lab. Also, you need to read the unknown project instructions file on Canvas for next week. Let me show you, I'm in here now. So you come down here and scroll down to week 5. Hopefully you can see that. Nope, I don't have it on yet. So let me turn it on right now. Those have to be off. Sorry, when I switch things, it switches everything on me. So now you can see week 5. And that's for the lecture, but let's come down to the lab. And that's the unknown test. Let's go ahead and show you that now. Where's the lab? There it is. So in week 5 lab, besides lab module 6, which will be at the start, there's a section I've got listed unknown report. You need to read the unknown instructions. And that'll be the instructions for doing your unknown report. And then we'll talk about that next week. Any questions? And now you're going to be overwhelmed because Lab 5 has a whole bunch of tests, but you don't need to read anything else, especially the Lab 5 unknown tests. Any questions? And I should probably turn on the lecture for all of the other weeks, but I'll do that after the lab. All right, any questions about what we're doing? No, I'm going to shut that down and come on to the lab. Sorry, I've got this in the way. Normally I have that set up there. I didn't have it set up. Sorry about that. So Lab 5, cultivation of bacteria, culture media, inoculation and isolation. Am I coming across okay?
Yeah, it's fine.
Thank you. So there's a little bit of reading, but not much to this lab. So pre-read pages 157 to 162 and 162 to 163. Culture media and obtaining pure cultures. There are some supplemental material in this lab, video clips, and a virtual lab. Your learning objections. Upon successfully completing this laboratory, you should be able to understand the importance of proper specimen collection and transport to the laboratory, be able to explain the 5 principal steps used by microbiologists to process specimens, differentiate between the various types of culture media, and explain the purpose of streak plate culture technique, meaning streaking for colony isolation. What's the purpose of that? And then define the following terms, as well as you should know how to do a streaking for colony isolation. Even though you're not going to do it, you'll watch a video on it. And then be able to define the terms selective media, differential media, and reducing media. Any questions about any of that? So in an introduction, the ability to isolate specimens that are pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, drying out, and exposure to oxygen, anaerobes in particular, and require special transport medium to preserve the specimen. Once a specimen is received by a laboratory for microbiological culture, microbiologists employ the 5 principal I steps to process it. First, inoculation, isolation, incubation, inspection, and then lastly, identification. Inoculation is placing the specimen on an artificial culture media to initiate growth. 2. Isolation, spatially separating microbial growth into distinct colonies. 3. Incubation, placing the culture media in an environmental conditions to facilitate their growth. And 4. Inspection, examining the culture for the presence of microbial growth. And then lastly, identification, performing specific tests to identify any pathogenic microbes that have been isolated. These 5 steps will be described, and they use just different types of culture media to get the work done. You should be familiar already with general purpose media, such as nutrient agar, Na, or nutrient agar with yeast extract, Na, or tryptic soy agar, TSA. These are general purpose media that generally just grow many organisms, but not all organisms. To promote the growth of fastidious microbes, those that have very specific nutritional requirements, we use enriched general purpose media. An example would be 5% sheep's blood incorporated into the growth medium to help organisms grow. And an example of that would be blood agar plates, or BAP. That's enriched general purpose media. There's also selective media. These contain chemicals that inhibit or halt the growth of some organisms. Some organisms can grow in the selective media and others cannot. Keep in mind 2 things when using selective media. First, a selective media isn't necessarily perfectly selective, meaning it may not select against all things 100%. And second, even those organisms that are supposed to grow in the selective media may form smaller colonies than growing in general purpose media. So the selective media usually isn't perfectly selective, meaning 100% selective. Then after selective media, there's differential media, and this media contains chemicals that produce visible differences among different groups of organisms when they're grown in that medium. So you can see differences between 2 different species. And these differences are unrelated to how well the organisms grow in the media. Any question about that? Lastly, the last media you need to know about is reducing media. This media promotes the growth of anaerobic bacteria because low or no oxygen conditions are required to grow these organisms. So we use reducing media to grow anaerobes because they can't grow in oxygen. It's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and a differential media. Here we're looking at McConkey's agar, and it's selective, and that is it allows gram-negatives to grow, but generally inhibits the growth of gram-positive organisms. So if you see growth on McConkey's, then it's likely to be gram-negative. It's differential in that it differentiates on the ability of the species that can ferment the sugar lactose. If the species grows, meaning it's gram-negative, and it can ferment the sugar lactose, it'll have a pink color to it. And it's pink just because when you ferment the sugar lactose, you produce acids, and this media has a pH indicator which turns pink when it is acidic. And it's differential that if the bacteria does not ferment the sugar lactose, it will be white or off-white. Okay? So McConkey's agar is both selective and differential. Blood agar plates are an enriched media, but they're also differential, and it can allow you to differentiate the different hemolytic patterns of the Streptococcus species. You can have no hemolysis, also called gamma hemolysis, shown here, where the media looks red all the way up to where this white is, and that's where the growth is. No hemolytic activity has happened. And then you can have alpha hemolysis, partial hemolysis. The white, once again, is the growth right here. And as you can see, it's red, but then it is dark or greenish right before the growth, and also inside the growth, meaning inside this alpha part. It's darker than here, and that's from partial hemolysis of the blood, giving it a dark or greenish color. And that's different from the beta hemolysis, which is complete hemolysis. Excuse me. It's red here, and as you come up to the growth, which is on this B here, the white area, that's the growth, we have the region that looks clear, and it's clear because all of the red blood cells have been completely hemolyzed. And that is beta hemolysis. Now you'll notice, usually around beta hemolysis, you have a ring that's dark, and that's a ring of alpha hemolysis. And so right in this region, there's no hemolysis. Right there, there's partial hemolysis. Right there, there's complete hemolysis. And usually when you're doing this reading, you don't look at where your growth is because you can't really see that area very well. But if you were to flip the plate over, you'd see underneath this there would be complete hemolysis, partial hemolysis, and no hemolysis. Any question about any of that? All right, so you should know this table. It's showing you the general purpose media, TSA, NA, and NA, promoting the growth of most organisms, but not all organisms. You will not get fastidious organisms to grow on these media, the general purpose media. Then there's enriched media like blood agar plates and chocolate agar plates. And blood agar is also differential, allowing you to see the differential hemolytic activity on the blood. The blood itself provides the nutrients to promote the growth of some fastidious organisms, and that's why it's enriched. Any question about any of that? The only difference between chocolate agar, CAP, and blood agar, BAP, is that the blood is added when the agar is very hot in the chocolate agar, and that causes the blood cells to lyse, and that gives the media a brown appearance that sort of reminded them somebody of chocolate, and that's why it's called chocolate agar. the blood in the blood agar plates is added when the blood has cooled, and I don't remember if it's 45 degrees to 50 degrees Fahrenheit. And is that right? Is that Celsius or is that Fahrenheit? I don't remember. Anyways, when it's 45 degrees to 50 degrees, and it isn't that hot and the blood doesn't lyse. Now 37 degrees Celsius is our temperature, so 45 would not be that much above, so it's probably Celsius, 45 to 50 degrees Celsius. All right, selective and differential media, and we've got 3 listed here: MAC, EMB, eosine methylene blue, and hectone agar, HE. They're selective and differential. All 3 of these are selective in that they select 4 gram-negative cells to grow, and they select against gram-positive. They're also differential, all 3 of them, on the basis of whether they can ferment the sugar lactose. And there'll be a different color if they ferment the sugar lactose. Then there's selective media. Phenoethyl alcohol is selective, and it selects against gram-negative cells, and so you use it to isolate gram-positive bacteria. Then there's selective and differential, which actually MAC is selective and differential, but mannitol salt agar is an example of selective. Oh, that's selective and differential. Never mind. Media, and this one is used to select for organisms that can grow in high salt concentration, and it's differential on their ability to ferment the sugar mannitol. All right, any question about any of this table? You might not have to memorize all of it, but you should memorize part of it, like certainly the general purpose, the differential and enriched media, selective and differential, selective media. Okay, so inoculation, that's something you do when you inoculate a culture. Isolation is something that Louis Pasteur developed when he came up with the concept of pure cultures. And he did have a way of isolating out cultures to get a pure culture, but his way wasn't very good. And Robert Koch developed the colony streak plate method, which was a much easier way to develop a pure culture. And we'll talk about this a little bit more. Streaking for colony isolation. You'll do that in this lab. When you're streaking for colony isolation, theoretically, in the last streak, or sometimes earlier, while you're streaking the plate, you'll reduce the number of bacteria that you plate. And the point is, someplace along your streaking, you'll reduce the cells that you're putting down to low enough numbers that they will form isolated colonies, as what happened here in the 4th quadrant. But it doesn't have to be in the 4th quadrant, it could be in the 3rd quadrant, which we were starting to get isolated colonies. Or maybe even one of the earlier quadrants. Doesn't matter when it happens, you just want to get isolated colonies. Maybe I should state what you're doing here is you're picking up on a loop bacteria and then streaking them out in quadrant 1, and then flaming your loop, and then picking up cells from quadrant 1. And you usually only do it 1, 2, 3 to 4 times. This is doing it 5 times. And then carrying from the 1st quadrant to the 2nd quadrant, carrying cells. And then once you streak that quadrant, you then flame your loop and streak the next quadrant, doing the same thing for all of the quadrants. Incubation, while you're culturing your media, they require chemical nutrients which are provided in the media, but they also require additional physical environment, and you can put them in different physical environments, such as growing them in certain oxygen concentration, in CO2 concentration, temperature, humidity, and obviously for a certain amount of time. Now, in our microbiology lab, our incubators only provide temperature and humidity as well as time. They do not provide specific amounts of oxygen and carbon dioxide. You then inspect the culture to make sure that there's obvious visible colony growth, and then look for differences. And then to identify the organism, you run relevant microbiological tests. And that would be identification. So you should know the terms of this lab. And in this lab, you will be watching a video on the inoculation of an agar plate using streak plate culture technique. You only need to watch that until 4 minutes and 15 seconds. This is using a 3-sector approach instead of the 4-sector quadrant approach, but both are valid. You'll also watch a video on beta hemolysis, alpha hemolysis, and gamma hemolysis, and this is done on a blood agar plate looking for hemolytic activity. And then you'll watch a video on McConkey's agar. You only need to watch it until 3 minutes and 50 seconds, and then you can stop the movie. And then there's a little video on mannitol salt agar. And then practice preparing a streak plate using the virtual lab. This does require Adobe Flash Player, and if you have trouble getting it to run, either speak to me or send me an email, and I'll try to figure out something for you to do. Okay? Hopefully it'll get to run. All right, and then work on your questions. Don't work on them here. Work on them in the worksheet, oops, which is shown here. Worksheet Lab 5. And that's due Saturday, January 30th, and it'll be worth 5 points. All right, I think that's it. Any questions? If not, it's about 7 o'clock. I'll be here until 8 o'clock to answer any questions. So go ahead and get started.